Hello, we cannot hear. Anybody who's on Zoom cannot hear.
Hello? Hello? Audience, are you there? Can you hear the speaker?
Yes, now yes.
Now, yes. Hello, Tobias.
Can you hear Raul?
Can you hear Raul?
Hola. Not important then.
Yes. No, we can hear. We can hear.
Okay,
now.
okay, hear
Everything.
Okay.
Okay, we're talking about university classes. I said Miguel Ángel Muñoz work on plenty on characterization of non-equilibrium university classes, and then he moved into more soft, I would say,
Soft.
like network theory and nowadays self-organized criticality, which is also kind of soft physics, and also into the neuronal dynamics. And he's going to tell us about this complexity of the microbial world.
Microbial world. Well, thank you, everybody. Thank you, Claudio, for inviting me, but Claudio is not around. Thank you, Claudio, wherever you are. It's always a real pleasure, and I really mean it to be here visiting you. By the way, the talk is in English, supposed to be in English. It's okay? Yeah. Real pleasure to be here because of the colleagues and also the young people and students and faces I know. So I really enjoy. Just I arrive and I feel happy. I want to talk to people and go for lunch and so on. And today, I have changed argument. Actually, in the last, I don't know how many, 5, 6 years, I've been talking about the brain, about neuroscience, essentially in every talk I was invited to give. And some of you, especially the oldest one, listened to my talk in recent conferences, either StatFis or in Barcelona at the Complex Systems Society. So I said, what the hell, I'm going to talk about something completely new. And during the Semana Santa break, it was raining a lot. I couldn't go out in the beach with my kids. So I said, okay, I'm going to prepare a new talk about something that I like, and I prepared this talk. So it's the first time I talk about microbes, and I feel I have a little bit of the imposter syndrome. So if there is any microbiologists in the audience, I really apologize. I'm going to tell you the things I learned in the recent 2 years or so that I got interested in the topic. We have also produced some papers, but I'm not here to tell you about my papers. I will at some point, but my main thing is to convince you that studying microbes is really fascinating for us complex systemists, right? So is the light okay? Maybe if I close a little bit. I think it's better like this, right? Okay, so I have a very longish introduction. Actually, if the whole talk is just introduction, it's okay with me. I'm not rushing to tell you my own results. And please, you can interrupt me, ask whatever. Maybe I don't know the answer, but you can try. And as I say, I'm going to talk about microbes, the things I learned about microbes. So look, this is the history of Earth. Well, Earth is born, let me see, where is the pointer? Okay, Earth is born here 4.2 billion years ago. And then we have all the way to go to humans appearing, Homo sapiens is appearing here. So essentially, we have never been in this planet, essentially the first approximation. But microbes appear really, really, really fast. So almost immediately after the Earth was here, it took very little time for microbes to appear, to start reproducing themselves, self-replicating and doing stuff. So first, they learn how to extract energy from different sources. Here is from hydrothermal energy. This is Yellowstone, as you know, and the different colors are different bacteria doing different things with different temperatures or different pH concentrations, right? So you have a community of different individuals doing different things, and they learn to colonize essentially all possible energy source. And very quickly, almost immediately after that, we have cyanobacteria appearing on Earth, and that was a big change because they learned how to use energy from the sun. They were pioneers in constructing solar panels, capturing photons and using also CO2 to produce, to fix carbon and to liberate oxygen. So after a few million years producing oxygen, they produced this thing that we now call atmosphere, which is full of oxygen and gave us space to develop ourselves and develop all life on Earth, right? If you take any piece of land, of soil, and you put it in this bottle, you can construct these Wiener-Gretzky columns in which you get that a long time. I mean, you put this in the bottle, you allow it to have some sun, some light, and then it develops automatically after months. These communities of bacteria doing different things. Here are more aerobic ones because you have some air entering here, some oxygen. Here you have anaerobic ones, and depending on the conditions on what you put in, you put some carbon source, some cellulose, or whatever. They survive. So it's a microcosm. They survive, and they have to take care of each other. So they create communities. It's not just individual strains of bacteria, species of bacteria. There is a large variety. One is doing something and something else, and they talk to each other and they exchange products, and whatever one eats is whatever one excretes is eaten by another one, and they construct these very nice and complex communities. Same thing in the oceans. You have also these microbial loops doing a lot of stuff. I'm not going to go into details what they do, but they are essential for recycling and for circulating energy in the oceans. So I put this quote from this gentleman here, Martin Blaser, who is a rock star of microbiome. It's a very well-known person. So this book of him, he said, they, microbes, made the oxygen we breathe, the soils we till, the food webs that support our oceans. Slowly, inexorably, through trial and error, across the deepness of time, they invented the complex and robust feedback systems that to this day support all life on Earth. Okay? Important feedback systems. I mean, this is not isolated microbes doing isolated stuff. They get together, they form consortia, they form networks, and they do this stuff. For example, this is a classical example for nitrogen. You have a cycle. So you have bacteria fixing nitrogen from the atmosphere, doing some nitrification, then nitrification, doing this thing that's essential for trees, and then this type, and then the nitrification. All this is controlled by bacteria and bacterial communities. Without bacteria, you kill bacteria here, and it's done. You lose the ecosystem, okay? You look everything. And here is the same thing, but for different things. Phosphate cycle, whatever, sulfate, whatever, metal absorption, degradation, many different things. So as I said, microbes colonize everything. And when more complex forms of life developed, that was another niche, an ecological niche to be colonized. And they, of course, colonized eukaryotes, so superior forms. And for example, here, they are in the roots, and they are in the leaves, and they're in the fruits, and they are doing different things, and they are in symbiosis with the plant. The plant by itself cannot do anything. It dies straight away. They need the microbes to do a lot of stuff, right? And they also colonize our human bodies. So this is a real picture of a colored picture of the tongue, some parts of the tongue. And each color represents a different type of phylum, of class of bacteria. Okay, so you see that there is a very complex community in the tongue, and as there is in every other part of the body, of the human body. What is more remarkable is that if you take the human body, you can count estimate, of course, the number of human cells, and it's like this number, 30 trillion, whatever it is. But now you count the number of bacteria you have within you, and it's 3 times larger. If you look in the internet, you will find a factor 10, but the most recent studies get it down to 3, 1.5, 2, 3, something like that. So for every human cell you have 2, 3 bacteria within you, okay? So now the word is holobionts, which means a whole thing, which is very inspiring for complex systems, right? It's the whole is much more than the sum of the parts. You have to take everything together, otherwise it doesn't work. And this is rather remarkable. So this is human cells, and this is, of course, this is a sketch, right? But this is even more striking. Here we have the number of genes, and humans we have 20,000, roughly 23,000 genes, depending on how you count. But we have 2 million genes, bacterial genes. So it's a factor of 1,000. So for every gene that you have that is processing some protein which is going to do something, you have 1,000 bacterial genes which are processing something and doing something possible. So the number of functions that these guys can do is overwhelmingly superior to our own. So we absolutely need them. Okay? And I like this analogy that I took from this book of this gentleman with physics. So essentially in physics, the standard model that we like very much explains like 5% of the matter in the universe. And then there are things we cannot explain, like dark energy, explain the expansion, accelerated expansion of the universe and dark matter, and that's the rest 95%. So in the case of biology, we have a similar situation. We have studied the human genes, but that's just normal genes. We have a whole dark matter of genes and functions and proteins coming not from our own genome, but from somewhere else, okay? So bacteria have diversified a lot, and you see the variety of different bacteria. This is a phylogenetic tree, is huge, and what you have here is archaea, which is another microorganism. So eukaryotes, us, it's nothing compared to the diversity of this world. And also if you look in total mass, so this is the total biomass in the planet, it's dominated by plants, of course, but all these are microorganisms, which is, I don't know, 1/4, 1/3, I don't know, no less. Well, whatever percentage, but compared to animals, it's much, much more, okay? And compare within animals, you have humans here, and elephants are here, right? So you think of biomass in the planet, and you think, oh, an elephant. The elephant is a negligible fraction. If you want to think in biomass, you think in the trees or in bacteria, okay? Oh. So coming back to humans, here you have this lady, where you have the different communities that, of course, depend where they live. They develop some assembling or some other. Again, this is a review paper of this Martin Blaser, the human microbiome at the interface of health and disease. And I would say that if there is a topic which is trendy now in medicine, collectively speaking, that's microbiome research. Okay, and what's the effect of these bacterial communities in everything? For example, here you have the first example. Here you see that in the stomach you have this small fraction. We all here have a small fraction of Helicobacter pylori, but this person here developed gastritis, and that's because this guy colonized the whole community, and then you have dysregulation, it's doing something else, producing too much acid, and then things start happening, right? But the origin is here. It's a bacteria doing something wrong. So I could be talking about this for long because I've been reading a lot about this, but just to give you some flavor, when you eat complex carbohydrates, so dark bread, not the white, ugly one, the real bread, you have complex carbohydrates that go directly to your microbiome in the gut because we are not able to digest them. So it's the microbes cutting them in pieces, and our microbes fermenting the fiber arriving there, and our microbes producing all the acids, short chain fatty acids, omega-3, omega-3, omega-6, all these things that are essential and that we are missing, and then we take supplements. If you give food, the right food to your microbes, they produce everything, but you have to feed them, right? Then, for example, in the gut microbiome in particular is essential for immune system regulation. So we have a barrier between our microbes, tons of microbes, and then ourselves. So there is this barrier, and the immune system is extremely present there because they have to control that microbes are not entering. So all the process of maturation, teaching the immune system what's yourself and what's microbes and what should come in, like all the things I said before, the omega-3, omega-4 of species, what has to come in and what cannot come in. So all that information is in the immune system and it's regulated in your gut, right? So if something goes wrong with the community there, you can start having problems related to immune systems like allergies or autoimmune diseases or immune-mediated diseases, right? Okay, many other things. This one, for example, all neurotransmitters are not produced in the gut by the microbes, but all the elements to produce neurotransmitters are produced by bacteria. So tryptophan, serotonin, all these things. If you don't have bacteria, you immediately stop producing neurotransmitters. Okay, and whatever, vitamin synthesis, everything. Okay. So this is the sad situation that we are living these days, especially in Western countries, where use of antibiotics has proliferated, and the use of antibiotics has saved tons of lives, soldiers in the war and these things, and infants at the hospitals, but has caused a tragedy in the microbial world. It's like bombing with atomic bombs our essential symbionts contributing to our health. So in Western countries, we have a lot of diseases, autoimmune disease, allergies, asthma, et cetera, that do not exist in Africa or in underdeveloped countries, and that essentially come from our diet, Western-like diet, and the excessive use of antibiotics. So once you have this regulation of the community of bacteria, you start running into problems. And for example, people with type 2 diabetes, they have this, systematically you can observe this, that, for example, this Clostridium grows up, and instead you have some other that goes down. And talking about functions, here you have a lot of transport of sugars, and then you don't have metabolism of vitamins, whatever. So you start running into problems. So take home message, they are crucial. There are a lot of things done by them, and without our microbes and microbes in a good shape, we run into deep shit. Okay. But if you look at the individual communities, you see a lot of variability. This is for the time being, this color is, each color is just a species, and this is across time. And what you see is that it's changing a lot. Okay. But instead, if you don't look at the particular species of microbes, but you look at the functions, what they are doing, the genes that are operating, you see something much more stable. For example, this is the number of genes producing devoted to fermentation, to oxygen respiration, to a carbon fixation, to whatever. So you see that it's very stable at the functional level. It's a machine, but it's very variable at the species level or the individual. This is very fluctuating, and this is very robust. And this is one of the things that we try to model and to understand, as I will show you. So it's very important. This one I added for Lucas, who asked me this morning. So it's very important to take care of your microbiome, and you can do different things. Something untargeted, so you don't have specific information about what's going on in your gut, but you do things like exercising or eating good things, or you can eat some communities of microbes. This is food for prebiotics, means food for your microbes in the gut. Probiotics means new microbes, like the ones you take with kefir and yogurt and these things, etc., etc. But you can also, these days, they are trying to do much more sophisticated things, like introduce new bacteria with specific genes that are supposed to do something in your community, or to put drugs in the community, or fake. And you can do a lot of things, as you can imagine. And this is a way to go in medicine, right? Okay, this is a bit like that. Fecal microbiota transplantation is a dramatic solution that works amazingly well. So this is just a shitty example, but I show it to you. So, for example, when you get very, very sick, you go to the hospital and they give you, because you have some infection, they give you antibiotics, and then you don't survive, they give you more antibiotics. Then you are cured of your heart infection, but your microbiome is gone. And then there are some opportunistic bacteria, like Clostridium difficile, that invades and take over the whole community. It's almost impossible to recover from that. And people, after a while with this infection, is diarrhea, and you die. Dehydrated and you die. Okay, so some fraction of people with some very small fraction of these people with Clostridium difficile infection survive, even more antibiotics. You kill also these, and then you're lucky and you recover. But now the standard therapy is fecal microbiome transplantation. That means that you take some popo from someone who is healthy, they prepare it, they put into your gut somehow. Don't try to do it at home, please. I'm not fostering these techniques at home, but they recover almost 100% of the times. Okay, so it's that you put a new a microbiome transplantation, and it works very, very fine. For the time being, only for a few diseases, especially for this infection with Clostridium difficile, but also for inflammatory bowel syndrome, síndrome de colon irritable. Now it's the standard therapy. And in the same way that you can recover or damage your microbiome, you can do that at a more environmental level. If you want to recover a forest after some tragedy, contamination, pollution, whatever, you have to restore the plant, you have to plant new trees, but that's not enough. You have to restore the microbiome. You have to take care of the soil and put microbial communities, and you have to do a full restoration. And these type of strategies are used now for pollution remediation. So you have oil spills aware, you can use, or people are trying to learn how to use bacteria to recover, to eat soil or whatever toxic you have produced and feed on that. Or for, this is used today for wastewater treatment and for energy production, whatever. Okay, so around this year, how I'm doing? Oh, as I promised, it's the longest introduction. So around this year, they launched the Human Genome Project to cartography all the genes we have. And about this 2003, the project was completed, and they said, okay, we have 20,000 genes, and we know all them, and we know there is very little variation among different populations, and essentially also no variation with respect to chimpanzees. 87% is the same as chimpanzees. But the story was not closed at all, because we have much, much larger differences than we see in our genome. And why is that? So the thing is that with all the technology developed to study our genes, some people, some clever people like this lady, Jo Handelsman, around the same time, started to study something called metagenomics, meaning that you go to the environment, like, for example, soil or ocean, you take a bunch of whatever, you take all the DNA, you amplify, you study the DNA, not of an individual, but of a whole community, of a whole ecosystem, okay? And you use essentially the same techniques. And this was this gentleman, Lederberg, who coined the term microbiome to refer to the set of genes in a community, okay? Usually there is a small difference. Some people talk about microbiota to the set of individuals, and microbiome is the set of genes. Okay? And for example, Craig Venter, who you know, was pioneer in sending an expedition, which is this one, the Voyages of the Sorcerer. You can read the book. Send an expedition to the ocean to study the microbiome and study the genes in the ocean in a generic way, as taking as much as possible and studying all. So we changed it from studying just the human genes to the metagenomics studies. And here is a summary of what you do. You take the environment, you take all the bacteria and whatever, you take the DNA, you amplify, and then you do a lot of bioinformatics that takes a long time, and supercomputers and all that. And at the end of the day, you have that in some community, like for example, if it's a gut, you have these relative abundances of species. a lot of this, very little of this, and so on, right? And you can reconstruct this type of time series. So this is one species, and you see in time how it goes, different species for different environments. Okay, so now that they have this technology, they launch a new ambitious program, which is the Human Microbiome Project, studying all the set of genes in our body, which are not humans, and also the Earth Microbiome Project. So we took advantage of this data. They have huge amounts of data, very high quality data, a long time, along individuals, people with disease, without disease, whatever, you name it. There is as much data as you want. And our idea as a statistical physicist was, is still to put together these two gentlemen. Okay, so do some statistical mechanics for complex networks like the ones generated by Microbial, right? So look, as a statistical physicist, I would like to look data and find statistical patterns that are preserved. I would like that. That would be nice, right? Like when you start to study gases, they discover a pattern, which is that pressures times volume equals K times temperature, right? This is a universal pattern, then you explain it. Then you propose a model that explains the pattern, and then you look when the model fails and what's lacking, you start adding ingredients to go ahead. Okay, so that would be the goal of the statistical mechanics of microbial communities. Okay, this seems physics, an argument of physics, but this is a book of macroecology. This is something invented by ecologists more than 30 years ago, and is doing the statistical mechanics of ecological communities. It's the same thing, the very, very same thing. And there are many, many papers in PNAS, Nature, Science about macroecology from microbiology. So this is from micro world to macro world, and patterns, scaling. There are many, many, many things. But what I want to start with, the second part of my talk, which is more going already to details, is started with this paper by my collaborator and friend, Jacopo Grilli. Some of you may know him. And he wrote this paper in Nature Communications, Macroecological Laws Describe Variation and Diversity of Microbial Communities. So he took the data I mentioned before from the human and other microbiome projects and produced this, look in the data, this type of time series, okay? So across time, how this species, its relative abundance in the community is changing. You have data, you can look at that, okay? And then you can look, as I said before, different species, and across time, see how they change. Actually, you can do two different types of studies. You can do this transversal, that means that your samples correspond to different individuals, okay? Or you can fix one individual and look across time how it's changing, okay? So I do both, or we do both along the study. And this is remarkable. This is the paper of Jacopo, and he found 3 super universal laws. This is more than universality class. It is only one universality class in the microbial world, and it's rather remarkable. So if you look, imagine, for example, this species. I think it's better to see it in the next slide. Yeah, I put this one here. You look at this species, and it goes up and down, up and down, up and down, fluctuates around some mean. So there, and this is the distribution of fluctuations. 0 is the mean, and it fluctuates, okay? You rescale all the different species you have, that can be 10,000. You rescale all of them, and they all collapse in the same plot, okay? So the fluctuations are a gamma distribution. The fluctuations around the mean. The mean, of course, is different for different species, okay? So how are the means distributed? That's this law.
The first one, the horizontal axis is the time or a given ecosystem, is the variation on time, or
both? I mean, in this case, is across individuals. So imagine here, I take a sample of all our guts, and I plot this species across people. And it's the same thing that to plot it across time.
Different people, or it would be the amount of that species in a given individual?
Both obey the same law.
Both have the same
statistics. Okay, so it's the same thing. The variability within me changing is the same thing if I do sample here and I say, okay, and it's a radic track if you want. If you want. Okay, so the fluctuations are a gamma distribution. And what about the means? The means are distributed as a log normal. So some species are extremely abundant and some are very rare, but most of them are very rare. That's a common trait in ecology. In ecology, it happens all the time, that you have the community, 90% of the individuals belong to one species, and then you have 1,000 species which are very rare, but that are there, right? It's the same thing here. And then we have Taylor's law relating the mean abundance with the variance of each species. So the variance is proportional to the mean squared. It's Taylor's law, another law which appears quite a lot in ecology. Okay, so the size of fluctuations depends on the mean. That recalls of what? Okay. I wanted Maxi to say multiplicative noise. Okay, so this is the ideal gas model invented by Jacopo in the same paper. He said, okay, I'm going to assume, as in the ideal gas, that species are independent. They don't talk to each other. And what every species is doing is obeying an equation, very simple equation like this. Stochastic differential equation. So it grows at some rate, which is here. Then it has to saturate at some point, right? So it limits this thing here is called in ecology the carrying capacity. And then essentially, I'm going to put a multiplicative noise, meaning that if you have a very large community, the fluctuations are going to be very large. Where is this coming from? This can be understood as a fluctuation of the growth rate. If you, in the growth rate here, imagine it's a constant, A. But then this A fluctuates, this translates into a multiplicative noise like this, as Max and Raul and everybody here knows very well. Okay? So the noise has to be multiplicative because reflects variation in the growth rate. And the growth rate is proportional to X. So the more you have, the more you can grow, etc., right? So these are the two ingredients that Jacopo proposed. And from this equation, you look at the station. You have a lunge event, you write the Fokker plan, exercise for people in the audience, master students. You write the Fokker-Planck equation in the Ito approximation, and you solve the stationary state, and you find this, and it's a gamma distribution. And if you assume that the noise is constant, you have the Taylor law, and if you assume whatever else, you have this. So here now I have the three laws for temporal data. Before it was for different samplings, now it's for temporal, and these are the prediction of the model, ideal gas model. Okay, with no interaction. Beautiful. So
What's your stage distribution?
the gamma distribution.
It's not model. Is zero is sufficient.
Series, but you have noise, and you are never fixed at zero. You fluctuate.
You reach zero.
You don't reach zero. That's the point. With the multiplicatives, with linear multiplicative zero noise, you don't reach zero. Okay, so you always fluctuate, and it's with square root you reach zero. With linear, you don't. Okay, so the model works perfectly fine, and I just say to say bravo to Jacopo. I really have the greatest of the admiration for him. He found data, he analyzed them, discovered universal patterns, and invented the mathematical models, very simple, with multiplicative noise, and explains everything. So, chapeau. Oh, very nice. Congratulations to Jacopo from here. And also, time-dependent properties are well explained by the model. Like, for example, if you have some abundance of some time, what's the abundance of some other time? And it goes linear, it should. I mean, you can explain more things, more patterns. I don't stop here. Okay? So, it's all the whole story. We have assumed independent species. Is there any evidence of correlations? Yes, of course. These are patterns of co-occurrence of microbes. So you look at different samples, and some microbes appear together, and some never. So for example, this guy always appear together with this and with that, or this cluster appear together. And if they don't appear, they don't. And red and green and red means two different set of patients. Actually, one are smokers, another are non-smokers here, whatever the community is. But the main thing is this. If you take the very same data that Jacopo analyzed, and you look at correlations, so what's the Pearson's correlation between two time series between two species, right? And what you get is these strange distributions here for 9 different communities. By the way, I didn't mention before that the different points with different colors correspond to different microbiomes. This is a gut, another gut, but this is an oral community. This is from soil, sludge, whatever it is, a glacier, a seawater, river, a lake. So it's very different. So it's really universal across many different things. And the correlations are also non-trivial. If the model was random, you still would see some correlations because of finite size effect. This would be the expectation for no interaction for independent species. But what you see is that there are much stronger correlations than you would expect if it was random. So the model, what's the model missing? The model is missing interactions. Okay? It's like an ideal gas. It works in many cases, explains many things, but not everything. So our first contribution to the field, a small contribution, was this paper. So we joined effort with Jacopo because I loved so much his paper that I want to do something with you. And he said, let's study correlations together. Okay, so with my PhD student, Matteo Sirecci, who did most of the work here, we went to study correlations. And to study correlations, we paid attention to phylogenetic distance, meaning the following. Which species are we going to look if they are correlated or they are not correlated? What we did is we take 2 species, for example, this one and these 2, and we can reconstruct, I mean, we use techniques from microbiome technology to reconstruct the phylogenetic tree and to learn how similar or how dissimilar they are. Okay, you can do that. I can explain you how, but I skip it. And now what we do is to find our own universal, my first universal pattern from data. I'm very proud of it. So this is phylogenetic distance. And this is correlation, average about all the pairs in the community. So you have species which are very, very similar phylogenetically, and they are here. And what we see in the data, this is looking data, is that they are correlated. So if 2 things are similar, they tend to be together. They tend to appear together. And as they get very, very dissimilar and they do completely different stuff, they get uncorrelated. You go to 0 correlation. But we have this pattern, and this here again, we have the same set of 8 different microbiomes. We have this universal decay of correlation with phylogenetic distance. Actually, we can quantify it as a stretch exponential, whatever. That's a detail. Yes. It's positive. Okay, so this is very strange. It's very, very strange for people working in ecology, in traditional ecology. Because in ecology, you have the principle of competitive exclusion. If you have 2 species doing exactly the same thing, 2 predators, I don't know, a wolf and something else, and one is stronger, more fit than the other, this one takes over and the other. And if there is a lot of one, there is few of the other. So you have anti-correlations, traditionally in ecology, coming from competition. Instead here, we don't. We have something else. So to study that, we propose our model in which we explain. I'm not going to go to the details of the model, but essentially it's a model in which we have equations for the abundance of the different species. Look at these beautiful plots for species. And on top of species, we are going to do something new with respect to the previous paper of Jacopo, which is putting explicitly the resources, what they are going to eat. Okay. So for example, if this guy and this guy happen to eat the same thing, they will be in competition. And we would expect that if you have a lot of this, you will have little of this. Okay, so negative correlations. And we are gonna let these guys fluctuate, meaning that we are going to give some rate of these resources, but it's variable in time. And on top of that, we also have another, what we call abiotic factors, like the pH, temperature, salinity, whatever. And these affect the growth of species, but there is no competition for these guys, okay? That's the difference between biotic and abiotic. if two guys of here eat of the same, they will compete for it. So this is a feedback loop. Instead here, not. They both can be interested in pH, but there is no interference from that. Okay, so here is the model. I don't want to annoy you with it, but it's the growth rate of a species and it's proportional to it. And this is the number of resources you have a type or another type. The important thing here is how we, let me go step back, how we specify a species in our model. And a species is identified by its preferences, what it likes to eat. So its preference in resource space. So this is a sketch for a D-dimensional space with all the resources, all the abundances of all the resources. And then you have your arrow saying, what I would really love is to eat glucose as a very high concentration every day. And every species has a slightly different preference for different things. So the similarity between two species is measured by this angle here, or the cosine of this angle. If the cosine is 0, then they are very dissimilar. If the cosine is 1, then they are very similar, right? Okay, so I go straight to the results. And the thing is that with this very general model, you can study a lot of things. Of course, you can imagine that depending on the rate I put the resources and how they fluctuate and how they couple, you can get many, many different things. So to keep it simple, we studied 3 different regimes that maybe it's better that I explain to you here. Okay, so in the first limiting case, what we have is that the resources here entering are fluctuating. And if there are a lot of them, for example, imagine that here, these 2 species like red, okay? They like eating red. So if there is a lot of red, they both grow. You see a lot of these guys, okay? And if there is less, there is less. This gives you to a flat correlation. This generates no correlation because, to give you some intuition, there are 2 competing effects. One effect is competition, I told you before. If there is a lot of a guy eating the same thing I eat, there is negative, right? But then at the same time, there is a positive effect. If the resource we both like is very abundant, we both grow, okay? If we both own glucose and there is tons of glucose, we both grow before we reach the limit of competition. So with these 2 things together, competing for resource and fluctuating resource, we don't get any pattern of correlation, okay? Then we have also this diagram here that, let me skip it, and I just go to the interesting one, just to go faster. Go to the interesting one. The one that reproduces the decay of correlations is the one which is dominated by fluctuations in the external resources. So how do I say this in a simple way that you understand it? I told you before about the model with species, which are with some abundances, and then resources, biotic, abiotic, whatever. To simplify things, you can integrate, forget about the resources, integrate them and put them into an effective equation, including only species, okay? And then the effect of resources and biotic and abiotic is reflected in some direct interaction here. So it's like a log-table terror type of equation. So you have here a matrix of couplings, and then you also get a correlation in the noises, okay? So our general model can be mapped in these different limits into different versions of this equation, where you have direct interactions here and indirect interactions through noise, okay? And the thing that is reproducing the empirical results is when these guys are not important, but when everything is controlled by environmental fluctuations, by noise, correlated noise, okay? So this is the empirical data, and this is what our model does. You say, oh, well, both are decaying, that's okay. If I select the right type of resources and fluctuations, I can reproduce empirical data. The only thing that's not right is that this is like this, and this is concave, and then the other is not working. The thing is that here, this is plotted as a function of phylogenetic distance, which is the distance in the phylogenetic tree. and mine is in the space of preferences, which is this cosine of these things. So we just have to map, and we make a prediction how some phylogenetic distance translates into a preference distance. And we have our prediction that has to be like some law. And once we put that, we get, this is our final model. So the final model is like the one with the coupled species, but now noise for different species are correlated. And the correlations depends on the preference for the species there. And when you do all this, we reproduce very well the empirical data. Okay. So look that the pattern we are explaining, this is the prediction of the model, and this is the empirical data, fitting just one parameter. Okay. It's very nice. The only thing I want to emphasize now for the last and second part of this is that the correlations are on average positive, but there are many negative correlations. Okay. So it's not that I mean, the pattern we have described is an average pattern. There are many, many fluctuations that are not captured by this simple discussion. So actually what I want to do in the second part, very briefly, I don't want to tire you, is to explain this variability in correlations with another simple model. You have a question?
For getting results, you still need all the biotic interactions? Or, I mean, you said that it's driven by all the abiotic fluctuations, but you need also the biotics.
You don't need them. You don't need them. I mean, the minimal model is without them.
That,
okay. So summary, the species, similar species fluctuate together. That's on average, but not always, right? But this is not the whole story. We have taken inspiration from this picture from physics, where you have different forces, a bit hot here. You have different forces in physics, right? And depending on the scale where you are, you can see more one force or another. So I strongly believe that this pattern that we are observing is because of the resolution of what we call a species that has some resolution. But if you work at another level, you could see in principle different things. Okay, this is just a side comment. Okay, so as I was saying, if you look in this part, you have that the average may be positive, but it's more frequent actually to be negative. So one thing is the mean, another thing is what's most likely. So this is for one of the microbiomes. This is the distribution of correlations, and all of them are peaked at negative values. The mean is positive because you have some very highly positively correlated species, but the most likely thing is to be negative, actually, and you have both. Okay, so this is the second part. For this part, with Mateo again, we joined efforts with a group of Jose Cuesta, who you know very well, in Madrid. We were interested in the same things, so we decided to work on these correlations together. And now the goal is to do the following. Again, we have the ideal gas, and we want to put something more, which is how the species interact between themselves. And that means a matrix of, in this case, we are modeling this just with the interactions here. And we want to infer, to see if we can infer the interactions between different species from data. But this, of course, is a very, very difficult problem. This is the inverse problem. If you give me a set of correlations and you want to infer interactions, that's an indirect problem. That's very, very tough, and you need a huge amount of data, and you never succeed at that. So what we decided to do is something slightly different, is this. If you look at the empirical data, you look at the distribution of correlations. So you have many negative correlations, many positive. If the community was random, it could be the blue one, but it's not random. So you have additional correlations. So the question is, how can we generate matrices, interaction matrices, okay, that put in our model, generate the actual distribution of correlations? So we are not modeling each pairwise correlation at a time, species I with J, no. We are just modeling on average all the distribution of correlations. Okay, the fact that we have a lot of positive, a lot of negative, and a mean that's negative. That's the thing we want to model, changing the interaction matrix. So we want to infer, this is the problem we have. This is the summary of the problem. We have some empirical patterns. You have to obey this, and you have to obey this, and the correlations like this, okay? And we have a Bayesian inference problem. Given the evidence of what you measure, which are all these patterns, you want to infer the matrix A in your model that it's able to do that. These are some technical details on how do we do it. But essentially what we do is Bayesian inference. We do a Monte Carlo algorithm. So you start with a random matrix, and then you try changes. And if the patterns reproduced by the new changes are better, describe better the real ones, you take it. If not, you refuse it, and you do this Metropolis algorithm. And eventually, you construct your ensemble of networks that reproduce very nicely all the patterns. These are the 3 patterns of Jacopo, and these are the correlations. So we have now an ensemble of matrices that we have analyzed, studied, that reproduces all the properties of the real microbiomes. And now the question for network scientists like Jose is, so how are these networks made? Okay. I myself have worked previously on ecological problems, understanding the properties of ecological networks. So I was really, really curious to see how are these networks of microbial. And the answer is, it's a mystery, because we analyzed a lot of properties, including the spectra. I promise unless to add some spectrum, a spectra of something. So we analyze all the properties, statistical properties of our ensemble are indistinguishable of a random network, but cannot be random. But we can random, you don't generate correlations. So there is something that is extremely hidden, because I've been thinking about it, and we with Jose Cuesta and all the co-authors for months, and we don't understand. We have a sub-ensemble of networks which have been evolved and reproduce all the patterns, but then you look at them and the statistical features are like random networks. I was very happy, said, no, the number of loops has to be different because loops control the field. No, identical. This is a random ensemble, this is our ensemble, identical. Then I said, no, the number of motifs, everything is identical, but they cannot be identical. So there is a puzzle here that next time you invite me at the IFIS, I hope to give you the solution. But for the present time, the only thing we know for sure, as this evolved network reproducing everything, are very sparse. So many, you start with the starting network where you full everything, it's a fully connected network, and then most of the interactions go to 0. The algorithm takes them to 0. And at the end, you're with a very, very sparse network, which still seems random, but reproduces all the patterns. And that means that essentially most of the interactions has zeros, are neutral. So most of the species do not talk to each other. The matrix is plenty of zeros. But some of them are very correlated and some of them are very highly anti-correlated. So in the last minute, I just tell you what we are doing now. So I joined Now Effort with my ex-PhD student, Juan Bonachela, who was my first student working on ecological problems. And now I'm very proud to say that he's a professor of ecology and evolutionary biology in Rutgers University in the States. And his neighbors are these 2 guys who are the rock stars of the microbiome. So Martin Blazer, the guy I mentioned a couple of times before, he has like 300,000 citations, I don't know. And he's a leader of the Human Microbiome Project. So we are very happy, excited to be working with them. And for the time being, we have not produced anything valuable, except for a bunch of Zoom meetings, trying to understand their language, because they are microbiologists. She, the wife and collaborator, is a medical doctor, and we are physicists. So, there are a bunch of barriers so they give us, we would like to understand this type of transit process because this is essential for the gut microbiome. What's this? Okay, so essentially you have some product here that's entering in the microbial community, and then here you have a second product, and at the end the final product is defined. Okay, so we have one product which is metabolized by some bacteria in S2, then S2 can be metabolized, and we construct this type of model. I think we are being pioneers in constructing a model of the microbiome where we have the species and not just resources as before. Many people are doing consumer resource model. We are doing a model in which you have the resources, but then the resource can be split in sub-products, which are the sheet of some metabolic process of someone, and are the food for someone else. So you have a whole change of whatever one is excreting is being eaten by another one. We are constructing the actual metabolic network and simulating that. And we are getting something, this is my last thing I say. We got something very exciting. I'm very excited about this, but still we don't have specific evidence. What we are seeing here is that our community, this is across time, and these are not species, these are functions. This resembles a plot I showed you at the beginning, that the functions are very stable. Species are extremely variable, are changing, new people arrive, they grow, they collapse, but functions, meaning, well, whatever they mean, functions remain rather stable. But here, suddenly, there is a collapse of the community and everything is taken by this guy. This happens spontaneously in the community, okay? And then the community is able to recover and regain again the functional diversity. So this resembles very, very much what happens to patients with Clostridium difficile infection, in which there is this species, Clostridium difficile, which is able to do everything. They have all possible genes. This is a generalist. And when they arrive and the community is weak, they take over and they do all the functions. So it's just one species doing everything, and then they collapse. And luckily, they recover without the need of a fecal transplant. They recover and go again. So our model is showing very interesting behavior where you can see the collapse of the community and the function and things. And we just want to push ahead this thing. I hope to tell you more news next time. So take home messages. Microbial communities are ubiquitous and fundamental for life. I hope I convince you of that. At least, if you want to take just one message, that's the key one. Species are not independent. They crucially interact between themselves and they create complex networks and complex ecosystems. The correlations are dominated by fluctuations in the environment, okay? And the interaction network are sparse, and there is a lot of work to be done. And it's a fantastic playground for statistical physicists, for complex systemists, because we have data, we have models, we have patterns, you can do everything, and you can have a lot of fun. And then it can even be useful for medical applications, which is also very exciting. So this is my collaborators and funding, and thank you very, very much for the attention.
Thank you very much for this talk. And any questions from the audience or from the Zoom community? Okay, Emilio.
Hello, nice talk. I'm sorry, this can look a very technical question, but I think is relevant. One is that you are completely neglecting demographic fluctuations.
Yes.
You only put external fluctuation in the environment. Just
Yes.
I want to see some comment on
Yes.
that. But also you have just mentioned, I don't know if this is true or not, that these fluctuations in the current in the growth rate, you put it in the ito sense. Has you said that?
Yes. Yes.
Can
Maybe
you justify or?
No, not really, but that doesn't change anything. That gives you only a shift in the mean, so it's really irrelevant.
Okay, and about the neglecting demographic fluctuations, you think
That's a very good point. The thing is that the size of these communities, when you have something which is abundant, is 10 to the 10. So the square root is 10 to the 5, compared to the multiplicative noise, which is 10 to the 10, is negligible. Of course, if you go to rare species, you should see some effect. So essentially what you do is you just neglect them, and species which are very, very, very rare, you essentially ignore them in the statistical analysis.
Other questions?
One question regarding the kind of interactions that you have. No, so you talk about the competition for resources. That's true. I mean, in ecological networks, that's one of the possibilities. But there is the other one, so which would be the mutualistic interactions. These ones that you are having this kind of network of
No, they are
of resources are related to the mutualistic, I guess.
No, it's
No?
everything. There are positive, negative, there's every possible sign.
Mhm.
You have the matrix, and every element of the matrix can evolve and take a final value. So you can have all these possible combinations. You have plus plus,
Mhm.
which is between I and J, and it's mutualistic. You can have minus minus and competition. Most of them are amensalism, meaning that one does not and the other is negative. I mean, most of them are neutral. They don't do anything. You have every possible ecological interaction.
Okay, so then you observe then when you are doing this distribution of the correlations, you observe the two kind of behaviors and the
Yeah, this is pairwise. You have everything. Now, if you do the distribution of the possible values, you have that. What I showed you here, for example. What is this? I'm sorry. Here you have
the
that
correlation
pairwise.
is-
You have positive, you have a
Mhm.
negative, and the most typical thing is negative.
Okay. No, because the positive will not be understandable otherwise. I mean, that's the
Yeah.
part. No, but for that they need to eat something close, not the same thing.
Yeah. No,
So-
probably the thing is that in these positive guys, what's happening is that one is something called cross-feeding. I have not talked about cross-feeding, but cross-feeding is the fact that one is excreting something that the other one guy eats.
Mhm.
So they go together.
Yeah.
The excretion of one is the niche, the ecological niche for the other. So the second one depends on the first. If the first one goes up, this goes up.
But in the last model that you have, when you have these products in chains, you are producing this kind of
Exactly.
of
In the
things,
last
no?
model, we have already this. In the first
Okay.
one, we don't.
Okay. Thanks.
I'm ignorant of how much of this is done at a different scale, but I mean, how universal is your universality? What I mean is, what you have at the end is a lot of Volterra equations with weakly coupled matrices or whatever, and multiplicative noise. Now, if you apply this same thing to, I don't know, the species of fish in the Mediterranean, how well does it work?
That's a good question. To be honest, I don't know. For example, the three laws that Jacopo discovered, the log normal distribution of abundances, that occurs all across the scale, and the Taylor law across everywhere. The gamma distribution, I don't know, but I think that they don't have data enough. The thing here is that you have 10 to the 10, so you can construct beautiful distributions. If you go to count elephants, you have 7 plus minus whatever, and it's so difficult. So the good thing here is that you have a lot of data, but I cannot answer your question how general that is. Also in macro ecology, usually you don't have, well, you also have cross-feeding. I'm not that sure. I mean, macroecology, you have some products and you can consume them or fight for them, be in competition. But it's not that that the guy eats whatever the other one eats at that largest scale, right?
Any other questions? If not, let's thank
Thank you.